I would start taking a point from the presentation of Professor Morton yesterday. Innovation in academia is 1% inspiration, 99% frustration, and an unaccounted for amount of persistent and fun. So let's try to have fun. I think you would agree that brain health is one of the big challenges of 21st century. And not only is affecting millions of people worldwide, but has been calculated by the European Academy of Neurology that the amount of money spent for treating them is, in Europe, 1.7 trillion euros. However, pharmacologically, the options are limited because of the presence of the blood-brain barrier that excludes 98% and over molecules from the central nervous system and almost 100% of biologicals. So in the quest for new therapeutics, the nose is an interesting venue because apart from being innervated by the trigeminal nerve on all its surface, the neuroepithelial olfactory region is the only part of the central nervous system that is in direct contact with the external environment. On the top of that, there is an anatomical proximity from the top of the nasal cavity and the central nervous system and the brain. It's not well understood how molecules can pass from the nose to the brain, but the most recent literature is pointing out that paracellular transport with perivascular, perineural, and lymphatic transport are to be accounted for this transport. The important thing is that there is a bypassing of the blood-brain barrier and, of course, is administration and a favorable brain-to-blood ratio. How are we developing products for this route? We are going towards classical methods that are already in use for nasal product, in particular starting with in vitro models of the mucosal tissue with primary nasal cells, established cell lines, and also more fancy tissue models like Uncilea. We have the possibility of using ex vivo nasal tissue, full thickness from animals like pigs, rabbits, or sheep, or from human nasal surgeries. We have the important casts and nasal models for the selection of the appropriate device, on which we will not speak too much today. And finally, I think the necessary use of in vivo models for the distribution to the central nervous system and with models of the disease, with the always present problem of how to deliver properly the formulation to the nose of these animals. We approached nose to brain many years ago, collaborating with virologists of the University of Bologna, because she told us that most viral infections are passing through an animal host, and when they are becoming more dangerous for humans, in most cases, 39%, there are neurological symptoms, and this viral encephalitis can be quite dangerous and life-threatening. And there are clinical approaches for this type of viral infections, like, for example, the use of antivirals systemically, like ribavirin, but this kind of high-dose intravenous administration of antivirals have many side effects. And so why not trying to deliver antivirals via the nose-to-brain approach? So as a lab, we are always walking the less common path. So instead of going for liquid formulation, we decided to do a little bit of particle engineering. At the time, we were developing this nice soft agglomerates called chimeral agglomerates. And we decided to produce particles with ribavirin microcrystals combined with microparticles obtained by spray drying, combining lecithin, that is giving the agglomeration capacity, with penetration enhancer that could help ribavirin to be absorbed nose to brain. And we selected mannitol as an hyperosmotic effect promoter, chitosan, mucoadhesive and tight junction opening, and alpha-cyclodextrin as a permeability enhancer of mucosal tissue. What we found, and first of all, we tried it in vitro with the mucosal tissue from rabbits, is that all the powder formulation were better compared to the solution, and ribavirin is highly soluble, 100 milligram per ml, but not all the penetration enhancers were equal, and not only the alpha-cyclodextrin was capable to provide an enhancement compared to the pure macronized crystals. Did it work in vivo? Actually, we were able, thanks to this collaboration with the veterinary faculty of Bologna, not only to look at the distribution in the brain, but at different regions of the brain. And in all the regions that we examined, the agglomerates were better compared to the simple powder or the solution. And we found the drug even at the cerebellum of the rats. In this case, suggesting a double transport olfactory and trigeminal. But most importantly, when compared to the intravenous administration of the same dose of the drug, we had comparable amounts in the olfactory bulb, but with a much better blood to brain ratio, so less side effects. So the take-home message from this first example is that hydrophilic drug, contrary to what could be expected, can be delivered nose to brain, and probably a powder formulation is going to be a right approach to this kind of molecules. However, we were in the heyday of nanoparticles, and myself, I was quite interested in testing this kind of vectors for nose-to-brain delivery. And of course, according to the nature of particles, you can expect different behavior. Positively charged particles are going to be mucoadhesive and probably interacting with the surface of the mucosa, while negative charged or neutral particles are going mucus penetrating and eventually can be uptaken. Actually, it's very important to know what kind of fate are going to have your nanoparticles, because, of course, if you are imagining that your nanoparticles are acting as vectors and are going to be uptaken by neurons or other cells in the central nervous system, you are going to have to answer some tricky question about the toxicity in this region of the materials that you are going to use. From our point of view, we went for the first hypothesis, more biodegradable and not so persistent, biopersistent particles. In particular, we used particles that were hybrid, constituted with lipids, phospholipids, and chitosan. And instead of going to look at classic, I would say, neuro therapeutics, we looked into a completely different class of drugs that were statins, because statins, despite being cholesterol lowering, they have a number of pleiotropic effects that are generally not visible because of the high metabolism in the liver. So using an alternative route like the nose to brain, we were expecting to find more anti-inflammatory, immunomodulatory, and even pro-apoptotic anti-cancer effects. So we developed these particles, and we also proved by small angle neuroscattering that they are interacting with some enzymes in the mucosal tissue like lysozyme or phospholipase A2 that are boosting the release at the level of the mucosa of the simvastatin loaded in the particles. Again, before going in vivo, we had a number of in vitro tests, and this is one of them with a human epithelium nasal model developed when I was in Australia with Paul Young and Daniela Traini. And this model is an air-liquid interface model with the possibility of, after 14 days, having a mucus layer and tight junctions in the particle. Comparing the nanoparticles, again, I would say suspension formulation, the improvement in the permeation across this barrier was several times the traditional formulation. So the nanoparticles actually improved the simvastatin permeation. So we were quite lucky to have two collaborations to test in two different models. For neuroinflammation, we worked with the Trinity College Dublin with the lab of Professor Kumlesh-Dev in a model of neuro damage and demyelination obtained with a neurotoxic compound that is psychosin. Actually, in this model, the cerebellar slices are stained with red to see the distribution of and activation of astrocyte, and in green with myelin. In the group treated with psychosin, there is a completely activation of the red astrocyte and the loss of the structure and the demyelination in the neurons, while the treatment with the neurotoxic compound and with nanoparticles prevented this kind of damage, proving that the nanoparticles could have a neuroprotective effect. But the most interesting result was for sure this work by our colleague of the University of Rio Grande do Sul in Brazil. We had a student in co-supervision, and the student tested simvastatin nanoparticles with chitos and mucoadhesive with a slight difference. Again, a model of glioblastoma, in particular orthotopic model of glioblastoma, and with a 2-week treatments with the nanoparticles at 60 micrograms per day, it was demonstrated that only the nanoparticle formulation was able to reduce the growth of the intracranial tumor, while the suspension of simvastatin had no effect on the growth of the tumor. Take-home message in this case is that when you have poorly soluble drugs and also problem of permeation, the use of enabling technologies like nanoparticles and lipid nanoparticles can be the solution for your approach. The last work that I presented from my lab is a post-COVID work that we did with cyclosporine. Actually, cyclosporine has a multiple type of effects, is a quite peculiar peptide, is poorly soluble, substrate of PGP, for sure is not going to the brain. So we said, why not trying for its anti-inflammatory characteristic with a different formulation, this time mucus penetrating, and we decided to use micelles. So we obtained ultra small micelles with a dimension below 15 nanometers, and we could check by small angle X-ray scattering that they have a quite fast diffusion in a mucus model. In particular, the rate of diffusion is 5 microns per second, and if you think that the thickness of the mucus is in the nose in the order of 10, 15 microns, it means that in 2-3 seconds, you have the saturation of the mucus layer. At the same time, we did also ex vivo nasal mucosal permeation with the rabbit mucosa, and there was a huge improvement of the permeability of the mucosa thanks to the effect of the surfactant that we used that was a derivative of vitamin E pegylated. So, do they work in vivo? In this case, we collaborated with the University of Coimbra, the lab of Professor Anna Fortuna, and they have a model of neuroinflammation obtained by injecting lipopolysaccharide into the mice. And they have a number of behavioral studies, so I'm presenting just one of the studies that were performed. And this is the tail suspension test that is assessing the capability of the mice to, I would say, react and try to escape the difficult situation of being pending from a bar. The mice with neuroinflammation were more, I would say, less fighting. They were more, I would say, were giving up early. The group with oral cyclosporine had no effect against this depression, while only the formulation with the micelles was able to recover to the control level of time spent in escaping behavior. We have other examples that I cannot present of micelles obtained with other peptides, and I can assure you that playing with the isoelectric point of these peptides, nice micelles can be obtained that are useful for nose-to-brain delivery. So again, when you have some biologics and you want to promote their nose-to-brain delivery, maybe the use of penetration enhancer in the form of micelles or other type of modulation could be quite interesting. So the last example that I'm presenting is not from our lab, even if we are starting working with RNA therapeutics, but is from the lab of Professor Tamara Minko at Rutgers University. And she had a nice couple of papers published last year about the administration of a triple therapy with memantine, donepezil, and siRNA against beta secretase for the treatment of Alzheimer in a model, genetic model of Alzheimer's disease that is suppressing amyloid precursor protein and presenilin 1. And it was possible to demonstrate that these lipoplexes had at least the same effect as memantine and donepezil, and the combination of the three drugs was actually reducing the amount of expression of beta secretase, so limiting the accumulation of beta amyloid, and again in a behavioral test could restore the spatial and orientation memory of the genetically modified mice. So very exciting. They found that the liposomes and the siRNA were co-localizing within the brain parenchyma in a much higher concentration compared to intravenous administration of the same type of delivery systems. Everything is very exciting. Thank God I'm on time, and I'm very proud of it. But there is a big chasm between the preclinical studies and the clinical development of a real nose-to-brain product. And there are several reasons for it. First of all, there are no real in vitro models of the neuroepithelium, and there are a lot of colleagues also in this audience that are working in co-cultures, organ-on-a-chip models, and other clever solutions for that. The ex vivo systems have not yet investigated deeply the possibility of using neuro-olfactory epithelium from animals and characterize properly how the formulation behave with this epithelium compared to the respiratory one. In vivo models are often in rodents, and as you can appreciate from the slide, there are huge differences in the nasal cavity and the neuroepithelial tissue between rodents and humans. The best model could be primates, but you are clearly aware of the ethical concerns about the use of primates. So one of the best shot could be to work with in silico models, especially in vitro, in vivo correlation and translational models able to translate the data with rodents to the human physiology. I hope that I convinced you with my 20 minutes presentation that it's an exciting time to work in nose-to-brain delivery. As always, I could not be here if not with the help of a lot of friends, and you could see from the collaboration I mentioned, and especially from young scientists that worked with me. And I thank you for the attention during these 20 minutes.
Excellent, Fabio. Thank you for taking all the efforts to keep on time and for still showing such impressive work. You're invited to ask questions. For us, it's easiest if you stand up if you want to ask a question, because then we can actually spot you, and the microphone persons can also spot you. So there is one over there. And while you get the microphone, maybe I can start with a quick question, because you showed a broad variety of different case studies where you show that it works, and you use various nano delivery systems. Could you tell which one works better? Is it a lipid-based or a polymeric-based, or what does it actually? Or what are the parameters you decide which to use?
Yeah, it's an excellent question, and the answer, as always from the formulator, is it depends on the drug you want to deliver. So I think that there is not one socks-fits-all type of solution. You have really to tailor your formulation on the drug, and the bigger the drug is from the point of view of the molecular weight, the more, in my opinion, you have to push with the penetration enhancing stuff. It's a trade-off, you know. It's always trying not to be too damaging the membrane, but opening it enough to get something bigger through. I like very much the micelle approach, because of the size and the capability of encapsulate peptides. However, the chitosan particles worked very well, and they have this I would say, degradable aspect that provides a real nice push for the permeation of the poorly soluble drug.
Thank you.
Hello, thank you for your presentation. I'm Purva Bhausa from Philips Medisize. My question is actually following up to the previous question, the micelles therapy or the micelles approach you showed. Do you see any hurdles in commercially scaling up that process?
You know, as I tried to warn during the presentation, I think that developing a nose-to-brain product, you have to be ready to answer questions about the biocompatibility and the eventual chronic damage of the formulation to the olfactory region and to the, I would say, part of the brain that could be interested by the presence of this material. It's a challenge, but it is one that you have to take if you want to have a project with high risk, high gain type of profile.
Is there another question? Yes, please.
Lovely talk, Fabio, and I think we're finally getting some excitement around nose to brain after a lot of skepticism, though I think that the evidence is still very small as to how it actually could be commercialized into a product. What do you think the type of bioavailabilities we could get from the nose to brain contribution of any product? Because we're going to get some contribution to brain effects from other means of targeting the brain, and other uptake, and I think the way to go is the in silico modeling of all of this. And I know that Will, has been looking at that in terms of in silico modeling. But what do you think it could be? The bioavailability, absolute maximum bioavailability for certain classes of BCS classification for nose to brain?
So it's an excellent question. And I think that we are talking about fractions and to be honest, 1% of the total bioavailability. What I'm stressing all the time whenever I'm talking about that is potency. We need to look into potent drugs that are going to have effects at very little concentration. One of the errors, if I can say so, of many of the literature that is present is to use drugs that are already getting into the brain, tested nose to brain, but that's make little sense. I think that we have to try to enable molecules that cannot get to the brain by other routes via nose to brain, and especially look at stuff like RNA therapeutics that can have potencies at minimal quantities delivered. Thank you very much for the question.
Okay, so thank you, Fabio, again for this very interesting talk.
Thank you, Regina.